In this video, we are going to observe um, osmosis using a potato as our sample organism. And so one of the things that I want to do is I want to make pieces of the potato that are of roughly equal mass, roughly equal volume, really roughly equal size overall, and they need to be a size to fit in one of our large test tubes. I need to record the mass of each of the potato pieces that I create, and I'm going to put them in specific test tubes so that they can be exposed to a specific concentration of solution over time. We're going to leave them for 1 day. So I'm going to start by coring my potato and making measurements of the mass of the potato in each of those solutions. So I've got a potato core right here. So the potato core is going to help me to puncture holes in my potato. And then if I need to cut them to equal size, I'm going to use a scalpel. It is sharp, so be careful. The potato core is pretty simple. You simply just sort of puncture through potato, pull it out, and if you need to, you can use a pencil or something to push the core out through the core. So I'm going to do that, and every time I make a core, I'm going to get the mass of it, record that on my sheet, and so keep that information available to you. So let's go ahead and get started. So I got my first core here. I'm going to push it out. Ta-da! And I'm going to keep making these until I have as many as I need. And I'm going to need 11 samples, so I'm going to want to do this 11 times. There. So I've cored my potatoes successfully, but I want these to be roughly equal volume and mass. So what I'm going to do is I'm going to cut the potatoes to remove any of the skin. It's one thing that I want to make sure that I do. The skin might interfere with how osmosis operates across the cells. And now I'm going to take the mass of the potatoes and I'm going to record the mass as I do that. And I've made a little data table for myself to record the number of grams. So let's start here. 2.82. And when I finish recording the mass, I'm going to put the mass here as well. And I'm going to add that to my first concentration test tube, which will be 0 molar. Let's do it again for the next one. Awesome. So I've cored my potato. I've made my potato into cores that are of roughly the same length, width, and mass, anywhere between 2.47 and 3.17. So we have a range of about 0.5 grams, which is okay for this particular lab. I'm going to clean up here, and then we are going to prep our solutions. Okay, so as you can see, I've got my test tube set up here. I've got my molar concentrations written here, and I've actually put the gram or the mass of the potatoes as they're associated with the solutions that I'm going to prep. I've also done my calculations here to see what my materials that I need are going to be. I have prepared a sucrose solution. So this is just sugar water. This is 1 molar sugar water. So I'll write that down for you. 1 molar sucrose solution. And what I want to do is I want to make 11 solutions between 1 molar, so this can go right into our solution, our test tube over here, and 0 molar. So how am I going to dilute this? Well, to have no molar sucrose solution, I'm going to just put distilled water into the 0 molar solution test tube, and then I'm going to dilute small amounts of sucrose solution getting larger as we go up the gradient, and small amounts of distilled water getting smaller as they go down the gradient. And I've got my calculations right here. Knowing how dilutions work, I can make sure that that happens. So we're actually going to start not with the sucrose solution, but we're going to start with this. This is our DI water. So remember, this should be 0 molar sucrose because it's just pure DI water. So we're going to start with this first, and I'm going to pipette it into the tubes down the up the gradient, and then I'm going to do the same thing with the sucrose solution. And I'm going to use a graduated pipette so I know exactly the volume of material that I'm using, or that I'm taking from our flask and putting into each of the test tubes. So let's start with DI water. So because 0 molar has no sucrose solution at all, we are going to just pipette 10 milliliters directly. Then we're going to do 9. In our 0.1 molar solution. So we are all done with DI water. And I should see a volume gradient down my solution, which I do. I can show you that on this camera. You can see the volume gradient dropping in volume all the way to no DI water. I am going to change pipettes because I don't want cross-contamination with my 1 molar sucrose solution. So now I've got a brand new pipette, and I'm going to do the same thing with my sucrose solution, but I'm going to go in the opposite direction. So we're going to start with 10 milliliters in the 1 molar solution. so now we are done with our sucrose solution. I have successfully pipetted that, and now we should be able to see that all volumes are the same. So let's make sure that all volumes are the same. We can eyeball it. They're not going to be perfectly exact, but we can see that the line of volume for each of the test tubes is roughly the same. So I am going to come back in 24 hours time. So we're going to wait exactly 24 hours, and then we will pull all of our potatoes out, and we will remass them, and we will see what the final mass is compared to the initial. So it's been 24 hours since we put our potatoes into our prepared dilutions of sucrose. Our 11 samples have had time to saturate in the solutions, and they've clearly changed over time. Here's an image of what they look like right now. What we need to do today, after the 24-hour period has passed, is we need to record the final mass of each of the samples that we have here. So what I'm going to do is I'm going to start with our 0 molar sample, and I'm going to go down the line and dump out the solution in the test tube, and then I'm going to use our scale to record the final mass. What we will do as a class is we will calculate the percent change of mass over time, and then we'll use that information to understand the osmotic relationship that's going on in the plant cells of the potato as they are exposed to concentrations of solution from the outside. So we'll be able to hopefully observe sort of a gradient of osmotic activity from very limited osmotic activity to maybe some higher osmotic activity going on at the ends. So let's see what we get. So we're going to start with our 0 molar solution. This is just pure DI water. So I am going to dump that out. I'm also going to pat down the potato a little bit, just to make sure that we're not measuring the water mass, we're measuring the true mass of the potato itself. Okay, here we go. 3.20. And we will do that for every other sample that we have. Alright, so we're all done. We've masked our potato samples. You can see that some gained mass, almost 0.4 mass here, and some lost mass, loss of about 0.7 here. So we're going to have to calculate the percent change, which we will do as a class in just a minute. One observation I want to make is that as we increased sucrose concentration in our solution, the material of the potato changed significantly. So this is our 0.1 molar solution of sucrose. And the potato itself is very squishy, very pliable. Feels almost like I could crush it between my fingers. It's extremely soft. And the 0 molar solution, this right here, is really tough. I can't break it. It feels really, really tough, like a hard potato. I would have to use a knife to break it. Yeah, I can't even do it with my fingers compared to the very, very squishy, noodle-like feeling of the higher sucrose concentration. And we get a gradient of that feeling moving along um, the lower sucrose concentration as we go. So that's one thing to note, that this is an observation that I made in person. I had to feel the potato to do that, and that's qualitative information that you might want to record for yourself somewhere. But now that that's done, we're going to go ahead and clean up, and we will work on finding the percent change in the data. And then we will work on looking at some similar data sets from other experiments just like this, and finding some statistical analyses of the data that we look at.